Okay, so now you know lots about the structure of membranes, we're going to move on to looking at how molecules move across them. Before we do, let's see how much you know about membranes. So can you pause the video here and write down as many things as you can about membranes, and then I'm going to review as many as I can think of, and when you're going to tick off to see how many you've got. Okay, so you should have rejoined me now. So what do you know about membranes? Let's think about the structure first of all. So what kind of molecules make up the majority of your cell membrane? Well, there's lots and lots of them. There we go. You can see them in this slide. So made up of many phospholipids. Have you got that in your key list? Maybe you've got some information about phospholipids. They're arranged in a bilayer so that their heads are facing outwards, because they're hydrophilic, and the hydrophobic tails are located inwards within the membrane, and that bilayer makes up the majority of the membrane. Okay, but then embedded in this membrane, you should also have that you've got lots of proteins. So we've got some proteins that go all the way through and actually provide a channel for the movement of molecules through the membrane. We've got other kinds of molecules as well that aren't just proteins. So we've got proteins with carbohydrate chains attached, and they're involved in cell recognition, and they're called glycoproteins. So within your membrane structure then, you labelled lots of different kinds of proteins and said different jobs that they had. So have you got a channel protein like this one? Have you got that they may be enzymes? Or they could be receptor proteins also in the membrane. Have you got that this whole structure is described as a fluid mosaic model? Have you said it's fluid? Because if you were to go back to the membrane, this picture, this membrane, a little bit later, we'd find all the proteins have moved position. Okay, so it's fluid. And also, because it's made up of all these different biological molecules, we say it's a mosaic. They're all different sizes and shapes, and they all fit together. Okay, so it's a fluid mosaic model. We've got phospholipids, that's the majority, arranged in a bilayer. Then we have proteins, lots of different kinds of proteins doing different jobs. And we've also got some glycoproteins involved in cell recognition. So some carbohydrate chains also present. And we do also, I'm just trying to spot one, there's one over here, have glycolipids as well, which is a carbohydrate chain attached to a phospholipid. And also in your cell structure, your cell membrane, or your membrane structure, you'll see within the membrane structure that you've got cholesterol slotted in between the tails of the phospholipids as well. And of course, that was about the fluidity of the membrane and the amount of cholesterol present. So I'm hoping you've got all those key facts, and maybe you've got some more. Brilliant work. Okay, so moving on then to our next task. Can you identify A, B, C, and D? Can you describe how these molecules are arranged within a cell membrane? And can you tell me which of them would test positive for the Biuret test? So pause the video here and have a go. Okay, so you should have started the video again, and we're going to review. So I would ask you first of all to identify what A is. Well, A is an example of a carrier protein. Okay, so it's a carrier protein. The evidence for this is that it's got a specific shape here to its binding sites, which allows a specific molecule to bind to that protein, and that protein will then carry, that's why it's a carrier protein, that specific molecule across the membrane. So that's that one. B, I'm hoping straight away, no problem at all, phospholipid. C, a little bit more tricky. So we've got protein with a carbohydrate chain attached. So that is one of our glycoproteins. Whereas D, we've got a phospholipid with a carbohydrate chain attached, which is a glycolipid. Okay, so glycoprotein, glycolipid. both involved in cell recognition. The carbohydrate chain sticks out from the surface, if you like, to allow recognition. So we'll meet them again as antigens when we think about cells being recognised in the immune system. So describe how these molecules are arranged. Well, the first mark point is going to be for the arrangement of the phospholipids in a bilayer. Okay, and then your second mark point is for describing that the other molecules are embedded within that phospholipid bilayer, creating a fluid mosaic model. Okay, and you can talk about proteins being present and being all the way through the cell, the phospholipid bilayer, or they might only be part way through. Obviously, if they're a carrier protein, they've got to go all the way through the phospholipid bilayer. So the challenge was which of the above would give a positive result with Bio-Ret test. So obviously we'd be adding our Bio-Ret reagent. It's light blue, which would test positive. Well, A and C, due to their peptide bonds within their protein structure, would give us a positive result, which would be a lilac or purple colour. Okay, so we said what we're going to do now is to think about how molecules move through this membrane structure. So in front of you can see a membrane. It's got the phospholipids in purple there, and you can see that's what makes up most of the membrane. You've also got some proteins present in that membrane. Okay, so let's introduce a type of molecule. Okay, so the type of molecule I'm going to introduce first is small, non-charged, lipid-soluble molecules. Things like carbon dioxide and oxygen. Well, because they are so small and because they have no charge, they are able to move through the phospholipid bilayer. So as long as there's a concentration gradient for them to move, okay, so there's a higher concentration of them on one side, they will move directly through the phospholipid bilayer. They don't need any help, any assistance in diffusing across the membrane. They just move through the phospholipid bilayer. Okay, so that's things like oxygen. I'm going to introduce another small molecule. Well, not molecule, small ions these would be. Okay, so if you've got small charged molecules, okay, things like just something simple like an ion, like a sodium plus, they have a charge. Okay, I'll try another one as well, like a chloride ion. Okay, they have a charge, and because they have a charge, they would not be able to move through the phospholipid bilayer. Okay, so they might try to, but they wouldn't be able to because of the charge. So they have to be facilitated, they have to be helped through the membrane, and it's proteins that do this. So here is a special kind of protein. Now these are specific to the ion that they transport, so they have to be the right size in terms of the channel and the right charges within that channel. So a sodium ion channel only lets sodium move through it. A chloride ion channel only lets chloride ions move through. So if you've got the right channel present, although it looks like it's slightly misaligned in my animation, it's supposed to be, they're all supposed to be going through this channel in the middle. The small charge molecules will move through that channel protein if it's present in the membrane. Again, there has to be a concentration gradient for that to happen. It's got to be more of those ions on one side of the membrane than the other. And if the right channel protein is present for that ion, it will then diffuse via the channel. Okay, so it's facilitated in its movement. Okay, so there's one other kind of protein that's required sometimes to help molecules through the membrane, and this one we met in our starter. Okay, it's called a specific carrier protein, and this works in a slightly different way. So let's take a look. Okay, so this molecule here represented is the sort of molecule that is a monomer unit. So things like glucose, fructose, any of the amino acids, they all have to pass through the membrane this way because they are too big to fit through the phospholipid bilayer and they're too big for any of the channels. So they need their own, very specific, and you can see very specific because it's got a binding site on the protein which binds to just this particular molecule that it carries across the membrane. Okay, so what happens when the molecule binds to its specific carrier protein? Well, it causes it to change shape. Okay, so the binding of the molecule causes the change in the tertiary structure of the carrier protein in a way that it actually moves the molecule across the membrane. And then the molecule is released and the carrier protein returns back to its normal tertiary structure, its original tertiary structure, and it's ready to carry another one of those molecules across. Okay, so these kinds of proteins work when there is a concentration gradient. They allow things like glucose and amino acids to move through the membrane if they're present down a concentration gradient. But as we'll see, they're also used if you need to actively transport molecules as well. Okay, but for that you need something extra, but we'll see that later on. So we've seen 3 different kinds of molecules really, and different ways that they can cross the membrane. Okay, so I'm going to now just remind you of something that you did in your GCSE studies, okay, which is the term diffusion. So we talked about how molecules cross the membrane, but this is really why they are crossing, okay? So diffusion, as you'll know from your GCSEs, is the movement of molecules from a region of high concentration to a region of low concentration through a partially permeable membrane. Now we're at A level, we need to think about the how a little bit more depending on the molecule. Okay, but the definition has not changed. Okay, I'm going to introduce this word before this kind of diffusion in a moment. Okay, so let's just remind ourselves of what diffusion is. Okay, so movement from a high to a low concentration. So what makes it simple diffusion then is, in this case, this molecule doesn't have a charge and it's very small. So it's moving directly through the phospholipid bilayer down that concentration gradient. So at A level we just say this is simple diffusion. Okay, it didn't need any help. So it's just through the phospholipid bilayer. So it's a passive process, okay, so it's not going to cost the cell any energy in the form of ATP because the molecules are moving from an area where there are lots of them, there's a high concentration, and they're moving down their concentration gradient. Okay, but it's simple diffusion because of the type of the molecule. In this case, we can think about something like oxygen. It's not charged and it's very small. Okay, but there were some molecules that we introduced that have to take a different route through the membrane. So now we have a different way of describing that kind of diffusion. Okay, so in both these 2 other ways that we've met, when you've got small water-soluble charged molecules and they move through channel proteins, things like I said, things like ions, okay, sodium ions, and molecules like monomer units, size molecules, things like glucose, amino acids. Both of them require help. They require facilitating. So they need a special protein present in the membrane for them to move by diffusion. And if that particular protein is not in the membrane, it doesn't matter if there's a concentration gradient for that molecule across that membrane because they can't get across. Okay, so there's still got to be a concentration gradient for it to be diffusion. Okay, but if the molecule is to move, so let's imagine we've got lots of sodium ions on this side of the membrane. for that sodium to move through that membrane, there must be a sodium channel protein present. And then that ion can move down its concentration gradient via the channel protein. Okay, same for something like glucose. Imagine now you've had your breakfast this morning, it's been hydrolyzed in your small intestine, you've got lots of glucose now, and you want to absorb it into your small intestinal cells. So lots and lots of glucose in the lumen of your gut. And we're hoping then that you're going to be absorbing it. So your epithelial cells, those cells that line your small intestine, will have absolutely loads of proteins, which are carrier proteins for glucose. So that when there's a high concentration of glucose outside of the cell, it can then move in via this specific carrier protein. Okay, so these two types of diffusion are still diffusion, so they're still movement of molecules from a high concentration to a low concentration, so you have to have that concentration for this to happen. But now you need another ingredient. You need to have the right protein to carry your molecule across. Still passive, okay? Now, thinking about that simple diffusion before, really, there isn't much of a limit to how many molecules can pass across the whole of the phospholipid bilayer, because there's a lot of it, okay? But one thing you've got to consider when you're thinking about these kinds of molecules, so things like ions and things like monomer units, glucose, amino acids, these molecules need to have the right protein to carry them across. If it's not present, they won't move, even if there's a diffusion gradient. And if it is present, how many of those proteins there are will determine how fast that can happen. Okay, so we'll see that in data sets, and we'll see that in our AO2, AO3 questions a little bit later on. Okay, so the rate at which this happens is really limited by the number of proteins, because they need to be present for facilitated diffusion to happen. Okay, now there's one last type of protein that has a really special job in the membrane, and these proteins are called aquaporins, and I bet you can guess already what they facilitate the movement of. They're specific to allowing water molecules to move across the membrane. Of course, water moves by osmosis, which really is a special kind of diffusion. Okay, so we'll consider it a little bit today, but then we'll have a little look at it on its own. But it's important to know that the water molecules need these channels to be able to cross the membrane too. Okay, but all membranes have them. Water moves across all membranes. So in the case of aquaporins, they're abundant. There's loads of them in membranes. Okay, so I would like you now to have a little think about the following. Okay, I've given you a list of molecules down this side of the slide, and I've given you a choice of 3 ways in which they can move. Now, all the time we're thinking about them moving by diffusion, or in water, a special case, movement down a water potential gradient, which is something we'll talk about later. So I just want to know how they cross. So if there is a diffusion gradient, how would they take, what route would they take across the membrane? Okay, so stop the video, copy it down, and record your answers, and then check back. Okay, so you should have completed the task, so let's do this together. Okay, oxygen. Well, I've mentioned it a few times. It's small, it has no charge, so you should have that it would move directly through the phospholipid bilayer, which is good because we want oxygen to diffuse through all membranes, which it will, and we want it to diffuse quickly. So because it's not facilitated by a protein, that means it's a really quick diffusion rate. Okay, what about water then? What about water? Well, we said water, particularly a channel protein, but specifically an aquaporin. Okay, so keep that one in mind. Sodium ions. Now it's really important when you're talking about ions that you either write them in the ion form, the chemist, or you make sure you have this word after them. Occasionally I forget to, you must always remind me to have that word ions there. So small but charged. So how will they get across? We'll need a channel to facilitate their diffusion. Okay, what about glucose? It's bigger now. So how's it going to get across? Specific carrier protein is what it requires. What about carbon dioxide? Okay, well that's small, doesn't have a charge. So just like oxygen, straight across the phospholipid bilayer. Again, rapid diffusion, doesn't need proteins. Chloride ions. Again, small but charged, so need a channel protein. Amino acids, they're bigger now, so they need a specific carrier protein, and that will be one for every of the 20 odd amino acids. Every one of them has a different specific carrier protein needed to take it across the membrane. And then finally, starch. Put that in on purpose to trick you, because of course starch is too big to move across a membrane, which is why we converted glucose to starch when we were storing the glucose in our plant cell, so that it would stay where it was formed. I'm hoping you've got all those right. If not, correct any mistakes you have and take some additional notes. Okay. So now I want to link to what we already can do. So I want you to think about the basic structure of an amino acid and draw it for me. And then I want you to use your diagram to explain why each amino acid needs its own carrier protein to diffuse across the membrane. Okay, so you can pause the video here and do that. Okay, you should be ready then to restart and draw, or you've drawn, so you're checking your amino acid. Okay, so our amino acid structure you need to be able to draw from scratch for the examiner. They are super keen. This has been on the exam for just 2 marks. I'm being very nice, giving you 3. Okay, so check off. Should we label it? So we've got the amino group. And then in yellow, our carboxylic or carboxylic acid group. Then, of course, we've got our central carbon with a hydrogen attached, and then this R is our variable group. So that's what we're going to use to explain our second answer. Okay, so each amino acid, let me get a mark point first of all, we're saying it has a different R group. Okay, so it's got a different structure in that region. Okay, so now if you think about that carrier protein, that site was called a binding site. It's really important you call it a binding site. Don't get it mistaken for an enzyme. Okay, so the binding site is going to be specific and complementary to that one amino acid. And that's because that one amino acid has that specific structure due to its R group. So only that one will fit. So only that one will fit into the binding site, and only that amino acid will cause that change in shape, that change in tertiary structure that we saw in the animation, resulting in that amino acid being carried across the membrane. So they're specific. All carrier proteins are specific for the one type of molecule that they carry across. And remember, these have got to be monomer unit size. Okay, anything bigger than that, it doesn't get carried across membranes. Okay, as we'll see in our next topic, digestion, we have to hydrolyze them down into those monomer units so we can move them across membranes if we want to. So far then we've learned about the basic structure of membranes, okay, and now we've learned that they're not always identical, that some of the proteins that are found in membranes are there so that we can move specific molecules across the membrane. Okay, so if you don't want to move glucose across the membrane, you just don't have a specific glucose carrier protein in your membrane. And then it doesn't matter if there's a concentration gradient, it can't move through the membrane anyway. Of course, this isn't the case with oxygen. Oxygen just diffuses across membranes, down concentration gradients, because it's moving through the phospholipid bilayer by simple diffusion. So quite a lot to think about there, okay? So how does your examiner ask you to apply your understanding of these two different types of diffusion? Okay, so let's have a look at this graph here. So which molecule, A or B, is transported into this cell by facilitated diffusion, do you think? Explain your answer. Stop the video now and have a go. Okay, so you should have picked B. And it's because of the plateau. Okay, so if you look at what's changed, the concentration of the molecule on the outside of the membrane has gone up. Now, you know from your GCSE studies, the higher the concentration gradient, or sorry, the steeper the concentration gradient, the faster the rate of diffusion normally. Okay, so the bigger the difference between the concentration on one side of the membrane and the other, the faster it is. And you can see that's certainly true for molecule A. As we've increased the concentration of the molecule on the outside of the cell, the rate it moves into the cell increases. Now you can probably explain that using your kinetic theory, can't you? So more of the molecule on the outside of the cell means it'll collide more often with the membrane. In the case of molecule A, it's moving across the membrane by simple diffusion, so just has to collide with a gap in the phospholipid bilayer and it can move across. Whereas at first, when you increase the concentration of the molecule outside the cell, for B, you do get an increase in the diffusion rate into the cell. But you get to this point here and it plateaus. So it doesn't matter that there's a higher concentration of that molecule on the outside of the cell. It's not moving any faster through that membrane. And that is, of course, because the rate has now become limited by the number of carrier proteins in the membrane. So it doesn't matter how much more concentrated the molecule is on the outside of the cell. There's only so many proteins that can facilitate its movement across the membrane, and they're working as fast as they can. All of the binding sites are full. Another way the examiner can ask about your understanding of membranes is a question like this. Explain why the rate of diffusion into the cell is higher for A than B. Okay, so they're both facilitated diffusion. I can see that because they both plateau, they both level off, even though the concentration of the molecule on the outside of the cell is going up. So what could explain it? Well, let's imagine the membrane structure, and let's imagine that there are lots more specific carrier proteins for molecule A, or more channels for A, than there are for B. The more proteins you have, the faster that diffusion rate will be. So quite often the examiner asks you questions with data, okay, to see whether you understand that all membranes are different, that although their basic structure is the same, they're mainly phospholipids, And then we've got a selection of different proteins that do different jobs for that membrane embedded in the phospholipid bilayer. If that membrane requires a molecule to move across it, then if that molecule is charged or it's a monomer unit, so it needs to be facilitated to be moved across, then that membrane has to have the right proteins to move the molecules that the membrane needs to have move across it. So membranes differ in the type of proteins that you find in their structure, which allows, when we do practical experiments like this one, where we're studying membranes, we can see differences in the rate of movement of molecules across membranes due to their structural difference. So in this case, molecule A, we've got more proteins to facilitate its movement, fewer proteins for molecule B, whether they're channel proteins or carrier proteins, which means that the facilitated diffusion of A is faster than B at the same concentration gradient, or with the same concentration gradient. Okay, so those last two are a bit tricky, so we will recap. Okay, so from your GCSEs then, list the factors that you think affect diffusion rate. So these don't include whether or not there's a protein present or not. So these are just other factors now outside of the ones that we've discussed. Okay, so pause the video and have a go. Okay, so we're going to have a go together. So we already said that if it's facilitated, diffusion, we'd have to think about, I'll write it in freehand like this. If facilitated diffusion, we have to think about the number of proteins. Okay. But generally now, if we're just thinking about all types of diffusion, so that's simple diffusion and facilitated diffusion, what factors affect it? So I'm hoping you've got things like the concentration on each side of the membrane. So we refer to this as the concentration difference. So how big's the difference between one side of the membrane and the other? The bigger the difference, the faster the rate of diffusion. Okay, what about the surface area of the membrane? Now, whether this is diffusion via simple diffusion by the phospholipid bilayer, or whether it is facilitated diffusion by proteins, the bigger the surface area of the membrane, the more protein carriers, more protein channels, the more space between phospholipids there would be. So the greater the membrane surface area in both cases, the faster the rate of reaction. And we met this already with the microvilli as a structural adaptation of the cell membrane. Okay, and also the cristae of the inner membrane and the mitochondria, again folded to give a large surface area. Now in that case it wasn't for the movement of molecules, it was for the presence of some other kinds of proteins that we'll meet next year. So surface area, concentration difference, and we've also got to consider how far the molecules are diffusing, so the thickness of the structure that they're diffusing across. When we do the lungs, I'm pretty sure you'll have done this when you did lungs at GCSE, we'll talk about the cells and how flat they are, so that the diffusion distance between one side of the gas exchange surface and the other is very, very thin. Okay, and the other one you might have come up with is temperature, which relates to kinetic energy. So increasing the temperature means we're increasing the kinetic energy of the molecules. They'll collide with the membrane more often there'll be more successful collisions either if it's simple diffusion with the phospholipid bilayer, or if it's facilitated diffusion with the molecule specific carrier protein or channel protein, so they'll move across quicker. So we'll just finish on that for now. So things that I've covered. So we've got a surface area, concentration difference, and also the thickness of the exchange surface. I did say temperature was another factor that would affect the rate of diffusion, which is common in practical experiments. But I thought we could have a go, just to finish up, with arranging these three factors together into an equation, which is called Fick's law. Okay, so which of them did we say had to be high to give us a good fast diffusion rate? Well, we said we wanted a large surface area, a large concentration difference, and we wanted a low or small thickness of the surface of exchange. So we'll meet this again when we look at gas exchange, because this is all about rates of diffusion. But in terms of arranging these in Fick's law states that the rate of diffusion is proportional to the two really that we need big go on top. So surface area times the concentration difference divided by the thickness of the exchange surface. So we'll be using this a lot when we look at gas exchange. We'll come back to that law. Okay. When we're thinking about membranes, it's the top two that are the most important. So how big is the surface area of the membrane, and what's the concentration difference on either side of the membrane? Both of those contribute to the speed of diffusion across the membrane. The thickness doesn't really come into it because membranes are generally the same thickness, 2 phospholipids across. But it's a good point to introduce this formula now for later. Okay, so you've now got a quiz based on this material, and by all means you can go back in the video and use the information to help you. Okay, so it's a forms quiz that follows this link as well. Thank you very much.